Going to hospital. Dress changing. In lab sample processing area. Incubator and hot air oven. Report entry area. Eating area. Microscopic zone. Machine for processing. Working in the lab. Doing gram staining first. Added crystal violet. Wait for 1 minute. Wash with water after 1 minute. Added grams iodine and wait for 1 minute. Wash with water after 1 minute. Added grams decolorizing reagent and wait for 5 seconds according to their capacity. Added safraning and wait for 1 minute. Wash with water after 1 minute. Let it air dry. Prepare food after done the processing of stains. Process stains observed under microscope. Pseudomonas organosa found. Work finished. We are ready for home. We are going to home.